as well as unsupervised learning in R. Please.
Thank you. Yeah, so this will be a very high-level talk, not so much about R specifically, but more about just ways to think about certain types of problems, including principal components, right? That was what it was built for, but I won't only talk about that. So that's the title of that. This is, yeah, so I'll talk about like thinking small about big problems. This is a kind of a mission I've been on for the last couple of years. So that's me, Bryan Lewis, and the work that I'll show you has been done together with these folks. Mike is at Yale, Jim is at University of Rhode Island, Lothar and Omar are local, they're at Kent State, and Carrie is at Johns Hopkins, and many others. But we work on what is best described as kind of efficient. One aspect of what we do is kind of thinking about problems and solving them efficiently, which is one aspect of what I'll talk about today. So it turns out that a lot of problems that people think are really huge are actually not as hard as they think they are. And my favorite approach, kind of my lifelong approach for solving these problems, is a technique called projection methods. And projection methods, we'll look at them in a very kind of tangible way in a moment. But they're good for at least two things. They help us seek solutions kind of in the right place. What does that mean? It means, it may mean a lot of things, but the most important aspect of that is it can reduce the influence of like noise or errors on solutions, right? And that's, another word for that is regularization. We're not going to, that's a whole talk in itself that we won't talk about. So instead I'll talk mostly about how to use kind of these projection methods to make huge problems that are really big and seem like they're really hard to solve smaller and easier to solve. So I like to say that projection methods turn big data into data. That's what they do. And one projection method that I'll talk about is principal components, but we'll talk about a few other things too. So kind of an easy way to start thinking about this problem is to really think literally about projection methods. So let's think about like a room like this that's full of people. And the problem is to kind of find people who are close together, like within some pre-specified distance of each other, right? So that's kind of a simple problem to state. It's easy to understand. And yeah, come on in.
This is the data science meetup?
Yeah.
All right, hey, nice to meet you. I'm Martin. All right, I'm just going to take a seat.
Martin, is it?
Martin, yes. Martin
okay.
Charles, like two first name things.
Yeah, so like we got this room full of people, and you want to. So one possible problem that you could state about such a room full of people is to find people close to each other. Really easy problem to think about. Projection methods will help us to make this problem simpler, although it seems really simple. They'll make it easier to solve this problem by ruling out people who are for sure far apart from each other, and then focusing only on the people who stand a chance of being close to each other. That's kind of the general gist of how a lot of this way of thinking works. So rule out as much as you can, and then solve a simpler, smaller, reduced problem. So let's say there's only 20 of us here, or less, but let's say there's 150 people in a room. Even with that few number of people, between every two people in the room, there are over 11,000 distances, right? So if you wanted to actually just measure the distances between everybody in the room with a ruler, you'd have to make 11,000 measurements between 150 people. That's a lot of measurements, right? And if there were 1 million people, or 81 million, or 1 billion people, it would be a lot more. It goes up, quadratically with the number of elements in the problem. However, if we project those people down to their shadows, like on a screen, like we're projecting here onto that screen, onto just a 1D line, you can sort all of those 150 people with only 750 comparisons. that's actually n log n instead of n squared. So it's a huge potential savings, and that savings grows dramatically with the number of people. So this is one kind of idea. So this is my kind of cheesy attempt at drawing a room full of people. There's not 150 there, but there's some people milling around in the room. They're kind of actually arranged not that differently than we are in this room. And so, for example, the purple person at the top and the pinkish person at the very bottom are obviously pretty far apart. They're not close to each other in that picture. So if we project, so this is my crude graphics. I drew this in R, by the way. So there I mentioned R now. So, this is like a projector, and I'm shining this light through these people and seeing where they lie on their shadows fall on that wall. Now, at this angle, the purple person and the pink person seem close together, so that's not very good, right? Because we know they're far apart. So what you want to do is you want to rotate your projection lamp around your data, the people there, until you get kind of the maximum spread of their shadows on the wall, right? So if we just swing that lamp around over here, now we've got like kind of the extremal 2 points there are as far apart as they're going to get, right? And in this, in this projection over here, we can see, we can't tell that much about these guys, right? They may be close, they may not be, we're not exactly sure. But we know for sure that these two are far apart. They have to be they're at least this far apart. They might be farther even. As long as the light and the wall are at a right angle with each other, we can say that just using kind of simple geometry. So we can rule those 2 points out. And similarly, we can rule that top kind of yellowish-green point and the blue point. We can rule some of them out, right? And then focus our attention on measuring just the ones that we're not sure about that might be close, right? That's how projection methods often work. That's all there is to it. This business of rotating the lamp until you get the maximum spread in the data, the shadows of the data, that's basically the idea behind principal components.
So where the light coming from defined by the maximum expression, or
Yeah.
is there any other way to define where the light will be?
Well, I didn't say it, but I'm assuming that I'm holding a right angle between the 1D line there that I'm projecting onto and the lamp that I'm swinging around, right? So and I'm only doing this, I'm going from 2 dimensions in this depiction down to 1. Obviously you can go from higher dimensions to lower dimensions, right? But here I'm just going from 2 to 1, and I'm keeping that at a right angle, the lamp and the screen. Now, the right angle is always part of principal components. There are a few other kind of details that we can skip over for the most part. I mean, this is the most important idea of principal components, that you're kind of finding, rotating your lamp around until your shadows have a maximum spread, right? You're looking for the point at which the data has maximal variability. And that's going to be the first principal component vector, actually. So that's kind of the idea behind principal components, looking for... And why do we do that? We do that because obviously, that projection doesn't tell us nearly as much as that one does, right? We just get more out of it, right? So we want to swing it around until we get the most variability there. And we still don't know everything about the data. We're losing information, of course, right? But we know as much as we can in 1D. And that's the idea of principal components. So that really simple idea goes a surprisingly long way. Like you can write papers about it, amazingly, which we did. And there's an R package that's not on CRAN yet, but there's a GitHub page. If you go to that GitHub page, there's an R package there, and then there's the reference to the paper that's not published yet. It's on archive, though, that describes this idea. And it's pretty amazing what you can do with it. So here's kind of a cooked up example. Since we're at the clinic, all of my examples are biological, but they apply to all kinds of fields. So I just took this has no biological meaning for those of you who are actually, geneticists or genomics researchers. But I just took a convenient data set. It's a big matrix. It's got 80 people and 394,000 measurements for each person, and they're measurements of their DNA this thing called methylation from the Cancer Genome Atlas, the TCGA, which is a big public repository of genomic information for cancer, curated by the NIH. And I said, okay, among all these methylated, 80 people with different genomic positions of methylation values, show me all of them that have a Pearson's correlation coefficient greater than some threshold, which is it's kind of like our room full of people problem. Show me all the people who are kind of close together. Show me all the vectors who are within a tight tolerance for correlation. And people actually do computations like this using brute force right now. I can point you to papers where they actually use kind of parallel brute force methods. And on this problem, the brute force computation takes 77 billion computations, right? It's a lot of correlation measurements because there's 394,000 columns. But that algorithm that we just mentioned, with the lamp, and ruling out people who are farther away and then focusing our attention on the close ones, that only takes 4 million evaluations. It's a lot less. So it's much, much easier. And that, the solution, which is described in that web page I showed you earlier in the paper, actually this is in the paper, not on the web page, it's pretty scalable. Like you can run it on this crappy Chromebook laptop, and it takes a long time, but it'll run. Or if you have a more powerful computer, it'll run in minutes or seconds, depending on how many cores you throw at it. It scales very nicely. Now that's kind of a toy problem, not so much a toy problem, but it doesn't have any necessarily any biological meaning. My colleagues and I are currently working on some new research that adapts this same kind of idea to these quantitative trait loci problems in genomics, the EQTL problem. And so we think that's going to be quite an interesting application of this, and it'll be really really high performance. Just, again, these are large problems that seem like they're hard to solve, but they're actually kind of easy if you kind of look at them in the right way and look for their solutions in the shadows. Yeah?
Are you looking for pairs of methylation locations, or are you looking for pairs of people?
Well, pairs of people would be very easy. There's not that many. There's only 80. So yeah, I'm looking across the 394,000.
I just want to find
all the pairs that are within that correlation threshold.
So you've got a bunch of points in 80-dimensional space, right?
Well, yes, each vector's got 80 entries, is 80-dimensional. That is correct, yes, yeah. And there are 394,000 of them, so it's a lot of possible pairs, 77 billion, in fact. So anyway, it's kind of a neat, real simple. Surprisingly, to my surprise, nobody's written about it before that I know until now. And then, so here, this is a pretty short talk. So this is the kind of the main topic. There's an algorithm by these guys, Jim Baglama and Lothar Reichel. Lothar is a professor at Kent State here in Ohio. Jim is at University of Rhode Island. And years ago, in 2006, they cooked up this algorithm called the implicitly restarted Lanczos bidiagonalization algorithm, or irlba, which is a wonderful acronym, right? Like everybody complains about the name of this, but it's a great it's actually a groundbreaking algorithm. I mean, it's truly very popular, very powerful algorithm, and it can very efficiently solve principal components problems, or kind of a related problem called truncated singular value decomposition for really large-scale problems, either dense problems or sparse problems, and either on a single machine or in a scalable way. So I first talked about this algorithm for the R language at the R. What are they called? The R meetings, the USAR conference in Dortmund in 2008. And it's been on CRAN for at least 5 or 6 years now. And it's a really popular package. It gets it's always between 60 or 70,000 downloads per month, actually. So it's a lot of people are using this package to do this. Unfortunately, we have a Python version, but it's not quite up to date with the R version. We'll be updating the Python version at some point. So Python's a little bit behind. There's really nothing else comparable to it in Python at the moment, other than our Python version written by Mike Kane at Yale and me. So here's kind of an example of the output applied to a biological problem. I took so variance, so again, another biological problem. There's this project at the National Institutes of Health called the 1,000 Genomes Project, and it's a misnamed project because right now there are 2,504 genomes in it. So it's the 2,504 Genomes Project. And they did full sequences very carefully for all of the subjects in the project, and they know everything about them. So it's kind of one of these things where you know a lot of information, whereas sometimes you don't know as much. These people all kind of signed away their privacy, I guess, for this project. And so what genomics folks do with genomes is they go through a very long process of measuring these. They rip apart your DNA with many replications. They measure it very carefully in these fancy machines, and then they kind of figure out, like a puzzle, where it actually belongs in your kind of DNA sequence. They put it all back together very carefully through many steps, right? And the last step of that is called the variants. The variants are like a human diff, like in computer lingo, if they measure my DNA and your DNA, and then looked at where they were different, and marked at every position, okay, there's a single nucleotide polymorphism, like you have an A and I have a G, right? Or I have some inserted piece of DNA that you don't have, or a deleted piece of DNA. And they do this for every position, all 3 billion positions on our genomes. And if we do that for these 2,500 people in the 1,000 Genomes Project, there are only, between all 2,500 people, there's only 81 million differences between them, right? So the number of variants there aren't that many, but there's 3 billion positions in our genome, and these 2,000 people, 2,500 people only differed in 81 million positions along their genomes. So what I did is I took all 2,500 people and all of their variants, all 81 million variants in this case, and that's a big, fat, sparse matrix, stored it as a sparse matrix in R and did a principal components on it, right? And plotted the first 3 principal component vectors. And this is what you see. If you just do it for a single chromosome, and I took a small chromosome, chromosome 20 is one of our smaller chromosomes in the number of base pairs, you get these really nice clusters of people, right? But they're kind of a little nebulous. But clearly there are differences. These clusters actually, there's the phenotypic information that the clusters correspond to, and these people are primarily their race actually. So there's like there are 2 Asian populations here. There's an African population, and there's non-Caucasian American population, and then there's a European Caucasian population. The interesting thing for me, and so you can see these types of plots a lot. This was just done in R using irlba on a single chromosome, and that problem I can run on this Chromebook in a few seconds, actually. But what you don't see often is that for the whole genome, all 81 million variants. And when you do it, there's quite a bit more fidelity. Like you can see, instead of these big amorphous blobs, there are clear subclusters in here, right? And so this is one of the blobs here that now has, maybe 3 subclusters in it. I don't know I haven't yet investigated, what the phenotypic differences are in those subclusters. But I just thought it was interesting that when you include all of the genetic information, you get a lot more interesting clustering going on there. And so this is another reason why we do projection, is to just visualize stuff. Because you can't look at 81 million dimensions, but you can look at 3 dimensions, right? And this algorithm lets you do that really easily on large-scale data. So it took me, on a 16-core PC to do the whole genome, it took 8 minutes with irlba, which is not bad, actually. And then the company I work for, we're putting code like this into publicly available containers that you can run anywhere. You can run them on supercomputers, or you can run them on your laptop, and to make it really easy to do things like this. So you don't even have to know much about R, actually, to run some of these containers. But yeah, anyway, so that's... so speed and visualization are another good reasons to do projection methods. And then I don't have too much more. I'm on slide 16 of 19. I wanted to mention an algorithm that came up. I was working with some guys at Novartis on a problem, and they were asking about, they love to do this thing called biclustering, where you kind of cluster the rows, you permute rows and columns of a matrix. So you put similar rows and similar columns together, and you end up with like these checkerboard patterns, right? That's biclustering. I don't have a picture of it. But it's a popular clustering algorithm, and but done in kind of 2D. And so there was a neat implementation by these two guys, Martin Sill and Sebastian Kaiser, at the German Cancer Research Center. And it popped up on my radar, and the Novartis guys were using it, but it was really slow. So we took a look at it, and it turns out that they were using projection methods. They only needed a low-rank singular value decomposition.
What's the SDV?
SVD stands for singular value decomposition. It's basically principal components. Okay. It more or less means principal components. It's principal components without the centering and the scaling. But other than that, it's like principal components. It's kind of the mathematical term underneath the idea behind principal components. Anyway, yeah, there's a lot of acronyms there, but these guys were using a specialized clustering algorithm that it turns out could benefit from projection, but the way they wrote it, they weren't using these clever projection methods. They were just kind of doing it brute force, right? And so these guys at Novartis and I were looking at it and we're like, oh man, this is actually a much easier problem. I mean, Novartis was running this problem in a kind of a non-clinical drug discovery setting. They were running it on like 500 node clusters, and it was taking like all day. And they were like, this has got to run faster. And so we took a look at the code and saw indeed that we could apply the same projection ideas to this algorithm, and lo and behold, we got it down. So here's just an example. Here's some R code. Here's an example using that package of a tiny randomly generated 1,000 by 10,000 example matrix. And the Novartis problems were at least 10 times larger than this. But the original algorithm took 3,500 seconds to run on a powerful machine. And by introducing these projection ideas that we've been talking about into the algorithm, we were solving exactly the same problem, but in only 20 seconds, right? So that was a huge reduction in time. And so Novartis was gracious enough to allow me to contact the authors so that we all work together to update the package on CRAN. And now everybody is benefiting from this performance increase when you use that package.
But conceptually, what's the difference?
It was the same as the thing with the at the beginning. They weren't using projection methods to efficiently focus the problem onto a small amount of data. They were just brute forcing the algorithm on a huge amount of data that they didn't have to. And all we did was introduce a few projections to cut down on the number of computations, but solve the same problem. And that's pretty much the trick behind a lot of projection methods. In the interest of time and boredom, I won't go into there are tons of other applications of these ideas. A bunch of people recently have applied them to some graph theory and network analysis problems, including this kind of network centrality problem, which does come up in genomics, actually, and social graph problems or fraud detection, counterterrorism applications. It comes up in a lot of applications. I've been working with those guys in the middle there on, they're mostly at Johns Hopkins on a social network problem application, these random dot product graphs. We just got a paper coming out. Well, it's in review right now. And then my friends down at Kent State and I are writing up some new grant proposals to extend these ideas to other network problems and kind of kernel methods and new sparse decompositions of problems for generalized linear models, other matrix decomposition, lots of application areas actually. So that we're working on now. So there's a lot of current research, and these problems. The great thing about this stuff is it's all very easy. There's conceptually, there's not. It's all very simple stuff, and it's very powerful. So there you go. That's my email if you want to ask me about anything, or if you want to fund my friends down at Kent State. They'd love to get your funding. Or if you want to use Entrepose to run your problems on supercomputers, you can talk to me too, and I'll put you in touch with the right people. But and now you've got some stuff that's much more practical than these high-level things, right? So thank you very much.
Thank you.